After a brief introduction of gene therapy, next is the types of gene therapy. In gene therapy introduction, we studied about what is gene therapy, the brief history of gene therapy, and the approaches of gene therapy, in which we studied about 4 types of gene therapy approaches: gene replacement or correction in case of a defective gene silencing in case of cancerous gene cells, gene augmentation in case the gene is missing, and suicidal gene therapy, that is again in the case of cancerous cells. So after these approaches of gene therapy, next is the types of gene therapy. Well, there are 2 types of gene therapy, that is the somatic gene therapy and germline gene therapy. As the name indicates, this somatic gene therapy targets the somatic cells. In this case, therapeutic genes are transferred into the somatic cell. In case of germline gene therapy, the therapeutic genes are transferred into the germ cells. The somatic cells that are targeted for this therapy, they are bone marrow cells, blood cells, and skin cells. And in case of germline gene therapy, the genes they are introduced into the egg and sperms. Next is obviously somatic therapy, it won't be heritable, and germline gene therapy being into the germ cells, it will be inheritable and will pass on to the daughter generations. All the research that is currently being done in the area of research gene therapy, that is targeting the somatic gene therapy. Okay, and for safety, ethical and technical reasons, this germline gene therapy, it is not being targeted. Now, after these types of gene therapy 2, somatic germline, this somatic gene therapy, it is further divided into 2 types, or we can say there are 2 methods or 2 approaches of somatic gene therapy: ex vivo, in vivo. As the name indicates, ex vivo gene therapy, it involves the transfer of gene into the patient cell outside the body, and in vivo gene therapy, the genes they are directly injected into the patient's body. So this is a diagram or a schematic diagram that is showing the gene therapy, both the type of gene therapy. So in ex vivo gene therapy, what happens? We take out the cell from the body and we culture them in vitro, the therapeutic gene that is cloned, put it into the cells, then transfected cells are there, they are selected, amplified, and they are returned to the patient's body. So ex vivo gene therapy, in which the gene was transferred into the cell outside the body of the patient. And in vivo gene therapy, this clone gene, it is directly injected into the body in combination, or like, it is not like directly gene is injected, it is put into some form of vector or a plasmid or a complex with the liposome lipid. Then it is put into the body of the patient, and then this gene goes to the target cell or tissue, and after this gene therapy showing the autologous, means the cells from the patient's body only, showing both type of strategies. So we will study in detail about these both types, ex vivo and in vivo gene therapy. Ex vivo gene therapy, it was its example is adenosine deaminase severe combined immunodeficiency. So this is the same disorder that this was the first gene therapy that was attempted with this disorder in 4-year-old that we have already studied. So what happens in ex vivo gene therapy? The genes that transferred to the cultured cells outside the body, transformed cells are selected, multiplied, and they are introduced into the patient's body. Mainly these are the cells, bone marrow cells and skin cells. They are the common targets, so candidate for ex vivo gene therapy. These are the cells which are taken out of the body. And after the establishment of human embryonic stem cell lines in 98 to 2002, these are also being, human embryonic stem cells, they are also being used for this strategy. Advantage is obviously being autologous cells, like the cells they are getting, or they're from the patient's body only, so it avoids immune rejection. Second is the transfer efficiency is high using retroviral vector, and transduced cells, they can be enriched using the selectable marker, means the transduced cells they are selected using this marker, And transfer efficiency, transfection efficiency or transfection efficiency, it can be assessed, evaluated before reimplantation of these cells into the patient's body. Disadvantage is mainly these therapy targets the mitotic cell, rapidly proliferating cell population is the target one. So tissue culture method is required. You should be having a proper in vitro culture condition for culturing of the cells outside the patient's body, and transplantation technology is required. That is how you are going to put back into the patient's body these cells. That technology should be clear and at hand before attempting this. So this is the diagram again showing the same ex vivo gene therapy. So as you can see in this, the cells, the cells, they are harvested from the patient's body that is defective in particular gene. Then that gene, that therapeutic, TG stands for therapeutic gene, it is packaged inside the retroviral genome, retrovirus, and this retrovirus, it is co-infected with the cells. So here the cells, they are taken from the patient's body, they are grown under in vitro condition, they are cultured under in vitro condition. Then these cultured cells, they are co-infected with the retrovirus that is already containing your therapeutic gene. Then the cells, they are selected, transfected cell or transduced cells, they are selected. It is not like every cell is going to take up the or every cell is getting infected with the retrovirus. There will be certain efficiency like 80%, 90%. So the cells, so we have to select the transfected cells from the non-transfected cells. So that will be based on the marker. So we will be selecting the transfected cells, and these transfected cells, they are further amplified, and then they are assessed for their transduction efficiency, and then they are put back into the patient's body. GM means genetically modified cell, they are introduced into the patient's body. The next therapy or next approach is in vivo gene therapy. For example, cystic fibrosis, it is targeting. So in this in vivo gene therapy, the cloned or therapeutic gene that is packaged into the vector, and this assembly, it is directly transferred into the target tissue in the patient through a syringe, or it is directly injected into the patient's body. And it is if possible, then this in vivo gene therapy, it is given preference over in vitro gene therapy. And it is done when it is done in vivo gene therapy when we do not have the culturing facility, in vitro culture facility for the target cell. For example, if brain cell is, if neurons are there, so we cannot culture neurons outside the patient's body, we don't have that facility still. So in that case we have to go for the other approach only. Or if there is no reimplantation technology, like how we are going to put back the cell into the patient, that technology we don't have for the relevant type cell or type of cell, then also we go for ex vivo gene therapy. So in this, gene needs to be delivered only in the target cell, and retrovirus, this could be the limitation of this therapy. Like we are using the retrovirus as vector, and they deliver gene only to the mitotic cell, dividing cell, proliferating cells. So it can target the cancerous cells. But in other cases, we are doing the improvements, like we can go for receptor mediated endocytosis, or we can use tissue specific promoters, like like to our therapeutic gene we can attach tissue specific promoters, so that these genes are expressed only and only in the target cells or tissues. And so this is how we can improve the in vivo gene therapy approaches. Okay, and the disadvantage is specificity is less, efficiency is less for stable gene integration into the genome of the concerned cell, and host immune response is there. If you are injecting DNA from outside, gene from outside the body, so there is always a chance of the rejection from the host immune system. So this is the diagrammatic representation of the same in this therapeutic gene, it is inserted, it is packaged into the virus or a liposome, it's a synthetic lipid bilayer, or a plasmid. And this genetically engineered DNA, it is inserted into the patient's body by direct tissue injection. I haven't drawn here a needle, injection syringe is drawn over here, and either of these complex, it is put directly into the patient's body, direct tissue injection, and the inserted DNA, it is incorporated into the specific cell or the specific target cell or tissue and produces the required protein that is encoded by this therapeutic genes. Okay, so this is the brief diagram. So we have studied about the two types of gene therapy, ex vivo gene therapy and in vivo gene therapy. What approach we are going to use, that will depend again upon the type of disease that we are handling. So sometimes one approach is better than the other. We can't say that this is better or that one is better. We need to decide depending on the type of disease. Next is, and the last topic from gene therapy is the vectors for gene therapy. So I'll be taking it in next clipping, but still I'll give you a brief idea. Vector for gene therapy, there are two types of vectors: viral vector, non-viral vector. In viral vector, these are the vectors: adenovirus, retrovirus, adeno-associated virus, lentiviruses, vaccinia virus, herpes simplex virus. So these are the common viruses which are used as vector for the introduction of therapeutic gene into the patient's body. Out of these first three, they are the preferred ones. And in non-viral vector, we go for lipid complexes to deliver our gene of interest or therapeutic gene. We go for liposomes, or we go for protein or peptide or polymer complexes, plus physical transfection method that may include electroporation and ballistic particle gun method. So these are the overall vectors that are used for introduction of therapeutic gene into the target cell, that I will be taking in next clipping.